A very good morning, students. And on behalf of NG Vartak English Medium High School and Junior College, I welcome you all for the new academic year 2021 2022. Students, last year we had studied full year online due to the pandemic, and as you know, still it is going on. So, this year also, we are going to continue with the online classes, with the digital classroom. I am Sarita Pandya teacher, and I will be teaching science to you. Students, before starting with the first science period of the new academic year, I wish you all a very happy and healthy academic year, and we all pray to God that you all come to school. We can have the physical classroom teaching learning session at the earliest. So, before starting with the lesson, let's all pray to God to have a healthy academic year ahead. Students, the very first lesson in your syllabus is Living World and Classification of Microbes. Living world. Living means all the biotic factors, all the living organisms. So in this lesson, we are going to study about the living organisms, and we are also going to study the classification of microbes. Microbes is not a new word for you. Microbes is originated from the word micro. Micro means very small, and the small organisms, the microorganisms, which we cannot see with our naked eyes, we have to observe them under the microscope. And what does a microscope do, students? A microscope enlarges the size of the object which is observed under the microscope. You all have seen the microscope, and it is used in the laboratory. As soon as we will start with the physical teaching learning session, as soon as you will start coming to the school, we are definitely will be going to the laboratory, and we will be observing all the instruments. We will be performing the experiments in the laboratory. We all are eagerly waiting to welcome you all in the school. So students, as I was talking about the microorganisms, the very small organisms which we cannot see with our naked eyes, and we observe them under the microscope. So in this lesson, we are going to study about the living organisms, the living world, and the classification of the microorganisms. So let's start with the lesson. Before we start with the classification, students, let's go through the history of classification in short. It was started in 1735. The scientist named Carl Linnaeus, he was the first one to divide all the living organisms into two main kingdoms, Vegetabilia and Animalia. Then in 1866, another scientist, Haeckel, he considered the classification into three. So he divided the living world into three kingdoms: Protista, plants and animals. But still, the scientists were not satisfied with this classification. And thus in 1925, Cheton, he created two groups. He divided the living organisms into two groups, prokaryotes and eukaryotes. In our 7th standard, we had studied about the prokaryotes and eukaryotes. And then in 1938, Copland, he divided the living organisms into four kingdoms: Monera, Protista, plants and animals. The word biodiversity. Bio, wherever the word bio comes, it means living. Diversity means the variety. That is, biodiversity means the variety in the living organisms. You know, when we see around us, whether the animals or the plants, there is a vast differences. There is a vast variety of plants and animals. If we look at the animals, some they live in the water, which are called aquatic. Some they live on the land, which are called as terrestrial. Some they fly in the air, which are called as aerial. Some live on the trees, which are called as arboreal. Similarly, the plants which are found in the water, they are aquatic. Terrestrial, found on the land. Some are amphibious. Amphi means both, they can be found on the ground as well as in the water. Then the animals, some are herbivorous, some are carnivorous, some are omnivorous, right? Similarly, the plants, some plants they can produce their own food, some plants cannot produce their own food. Then the size and shape of the animals, plants, they have a vast variety. So this is all nothing but the biodiversity. So the different variety found in the living organisms is called as the biodiversity. So why the need of classification was arise? Now, as I said, there is a vast biodiversity. There is a variety of living organisms around us. Now, it is not possible to study each and every organism in separately. So, to study the living organisms, it was very essential to divide them into groups. So, the groups and the subgroups were created on the basis of their similarities and the differences. So, the process of dividing the living organisms into groups and subgroups on the basis of their similarities and differences is called as biological classification. Why it is called as biological classification? Because we are classifying the living organisms. As I told you, students, bio, wherever the word bio comes, it means living. Okay, just 1 minute. What is biology? Biology is the subject in which we study about the living organisms, right? This is just for your information, and biology is divided into two branches, botany and zoology. Botany is all about plants and zoology is all about the animals. So let's again come back to the lesson. Biological classification, because the classification is done for the living organisms, thus it is called as biological classification. The scientist Robert Whittaker, he was an American ecologist, and he divided the living organisms into 5 groups, and on the basis of certain points he gave the classification. He divided these organisms into 5 groups. Now, what were the points? What was the criteria for the classification? So they were, first is complexity of cell structure. That is whether the organism is prokaryotic or eukaryotic. We had studied prokaryotic is the primitive cell, whereas eukaryotic is the advanced cell. Then the second point is complexity of organisms, whether the organism is unicellular, that means it is made up of one single cell, or more than one cell, that is multicellular. Third was mode of nutrition. Nutrition is to have the food. So whether they are autotrophic, auto means self, whether they prepare their own food, heterotrophic, whether they are dependent upon other organisms for their food, and saprophytic means whether they absorb their food from the dead organisms. So this was third point, mode of nutrition. Fourth one, lifestyle, whether they are producers, that is they can produce their own food, consumers, they have the food from other animals, or decomposers, whether they decompose the complex dead bodies of the animals into simple substances so that they can again mix in the soil. And the fifth one is phylogenetic relationship, that is from prokaryotic to eukaryotic and unicellular to multicellular. So these were the 5 criteria, 5 points on the basis of which the classification was done by Robert Whittaker, sir. So let's start with the very first kingdom, that is Monera. I hope you remember I had given you the classification that Whittaker divided the organisms into 5 groups. So the very first one is Monera. Now this Monera, it is the first group or the first kingdom. Now the organisms which are kept in this kingdom are unicellular. Uni means one. So these organisms are made up of only one cell. They are autotrophic or heterotrophic. Autotrophic, as I said, they can prepare their own food. They can synthesize their own food. Or heterotrophic, that is they are dependent upon other organisms for their food. Third one is prokaryotic, cells without distinct nucleus or cell organelles. As they are unicellular, so they are prokaryotic, in which the nucleus does not have a membrane, as well as the cell organelles also do not have the cell membrane. And the examples are Clostridium tetani, Salmonella typhi, Streptococcus pneumoniae and Vibrio cholerae. So these are all the examples of the kingdom Monera. The next kingdom is Protista. Now these organisms, they are also single-celled organisms, but they are little advanced from the Monera. They have a very well-defined nucleus. That is, they have the nucleus which is enclosed in a nuclear membrane. In Monera, the nuclear membrane was not present, but as the Protista kingdom is bit advanced from Monera, so here the nucleus of the single-celled organisms are having a distinct nuclear membrane. Then second is they have pseudopodia or hair-like cilia or whip, which helps in the locomotion. These are all the different adaptations for locomotion. Pseudopodia is made up of two words. Pseudo means false and podia means foot. So they have a false foot to locomote from one place to another. Or they may have the hair-like cilia, as you can see in the euglena, right? Or a whip which is used for locomotion. See students, on the screen you can see amoeba, and you know that amoeba does not have a definite shape, right? So as it locomotes, as it moves from one place to another, it protrudes a part of its body forward which forms a foot-like structure. Thus it is called as pseudopodia. Okay. The third one is they may be autotrophs like euglena or volvox, or they may be heterotrophs like amoeba or paramecium. Now volvox and euglena, they are autotrophs. They can synthesize their own food because they have chlorophyll in them, and thus they can produce their own food. So this is all about protista, and the examples, as I had told you, volvox, euglena, they are autotrophs, paramecium and amoeba, they are heterotrophs. Okay, let's move on to next. The third kingdom is fungi. Now, can you tell me very often where you find this fungi? Yes, you find it in your kitchen. And if you have not seen it, you can perform a small experiment. It's not an experiment, rather you have to take a piece of bread or a piece of chapati and you have to keep it open in the kitchen for 3 to 4 days. As it is rainy season, within a day or two also you could see a yellowish green color spongy structure on the bread or chapati. Even if we store papads or chips for long time, then due to humidity this fungus grows on the papad or on the chips. So this is fungus. Now the fungi, it is non-green. It is not having green color, so it is saprotrophic. It absorbs the food from decaying organic matter, right? So it is non-green. It is eukaryotic, right? That means it is having a distinct nuclear membrane. It is heterotrophic. It cannot prepare its own food, and most of them are saprotrophs. That means they feed upon the dead, decaying organic matter. They have a cell wall which is very tough, very strong, very hard, and it is made up of a complex sugar which is called as chitin. Okay, so the cell wall is made up of chitin. Some of the fungi are thread-like and they may have many nuclei present in the cytoplasm. Students, we had studied the structure of cell in 7th standard, and we know that cytoplasm is the liquid, is the fluid present inside the cell wall or the plasma membrane, in which the cell organelles are embedded, right? So in case of fungi, this cell wall is made up of chitin, right? And the examples are aspergillus, which grows on corn, then penicillium, mushrooms. Okay, as you can see in the picture, the labeling is given, though it is not there in the textbook, but this is very commonly found in our kitchen. As I told you, that it grows at a very faster rate during the rainy season because the climate is humid. Okay, so this is all about the saprophytes or the fungi. The next topic of the lesson is classification of microbes or microorganisms. So, in this part of the lesson, we are going to study about the classification of microbes or classification of microorganisms. Now, the microorganisms are mainly divided into two groups, that is prokaryotes and eukaryotes. Prokaryotes are further grouped as bacteria and eukaryotes are further classified into protista, algae and fungi. Be studying the size of the microorganisms, we have to study them in units of micrometer and nanometer. And 1 meter is equal to 10 raised to power 6 micrometer and 1 meter is equal to 10 raised to power 9 nanometer. So these are the this is the classification of microorganisms. So let's start with the classification of microbes, and the very first one is prokaryotes, which has the group called as bacteria. Now these bacteria, these microorganisms, these prokaryotes are having a size from 1 micrometer to 10 micrometer. Now 1 micrometer is also called as micron and it is equal to 1 into 10 raised to power minus 6 meter. That is 1 millionth part of a meter. That is 1 micron. Now these bacteria or these prokaryotes, they are unicellular, that is they are made up of only one single cell. They are independent, they are not dependent, they are independent, and sometimes they are found in colonies. So either they are found single or they are found in colonies. Cell wall is present, but there is no very distinct nucleus or cell organelles are present, as they are very primitive cells, as they are prokaryotes. And we had studied that prokaryotes, they do not have a clear nuclear membrane and the cell organelles are also not having the membrane. Then they reproduce by simple binary fission. So 1 divides into 2, 2 divides into 4, right? Then they are very vigorous in growth. So when they have a favorable condition, they grow at a very faster speed and they can just double their number in a time limit of 20 minutes, right? And there are different types of bacteria, as you can see on the screen. They are coccus, that is round shape. They are cocobacillus, they are spirillum, they are bacillus, they are vibro or comma shaped, they are single, or they are found in a colony of say tetrad or diplococci. Diplo means 2, tetrad means 4, right? So this is all about the bacteria or the very first prokaryotes. The second one is protozoa. Now protozoa, they are approximately 200 micrometers in size. They are either found in the soil or in the fresh water or in the sea water. They can be found anywhere and they are also eukaryotic unicellular organisms. They are also not having distinct nuclear membrane, and they are also made up of single cell. There is a vast variation in their cell structure, their modes or organs of locomotion, that is movement from one place to another, and they even have the different modes of nutrition. There is a great variety in them, and they also reproduce by simple cell division. A cell divides into two, and the common example of protozoa is ant amoeba, histolytica, then Plasmodium vivax, that is also a protozoan. Then Euglena is also a protozoan, and it is autotrophic, right? In the previous slide, we had seen that they can be autotrophic or they can be heterotrophic. The third one is fungi. We had already studied about fungi, so we'll just revise it. Their size is from 10 micrometer to 100 micrometer. They feed upon the dead organic matter or dead bodies of plants and animals. So they are also called as decomposers. They decompose the dead bodies. They are also eukaryotic in structure, that is, they do not have a distinct nuclear membrane, and the organelles also do not have the membrane around them. They are made up of just one single cell, but they are little bigger in size and they are visible with our naked eye. They are saprotrophic, as I said. They depend upon the dead organic matter for nutrition. They reproduce sexually as well as asexually. And when they reproduce asexually, they either reproduce by cell division or budding. That is, the buds are formed, and then as they mature, the buds, they get separated from the parent body and they develop into a new individual. The fourth one is algae. Now, the algae are also called as autotrophic organisms because they synthesize their own food as they have chlorophyll. They are aquatic in nature, that is, they are found in water. They are also eukaryotic, unicellular, and they are autotrophic. They are autotrophic, thus they synthesize their own food during the process of photosynthesis, right? And the examples are chlorella and chlamydomonas. Now here some of the algae are unicellular, whereas some are multicellular, which we can see with our naked eyes. Okay. As you can see, the algae, chlamydomonas and chlorella, they are green in color because they have chlorophyll. Okay. The last one is virus. Now, virus, they are having a size of approximately 10 nanometer to 100 nanometer. Now, actually, these are said to be the organisms at the edge of living and non-living, because they are not considered as living organisms, but as soon as they enter any living cell, they become living, right? They are very, very small. They are extremely minute, and they can only be seen under an electron microscope. Electron microscope is more advanced than the normal microscope, and because these viruses are extremely small, very, very microscopic, and thus they can be only observed under the electron microscope. They are a form of independent bodies. That is, they are made up of either DNA, that is deoxyribose nucleic acid, or RNA, that is ribose nucleic acid. There is a long molecule of DNA or RNA and it is covered by a protein coating. This is the structure about a virus. Now these viruses, they cause many diseases in the plants as well as in the animals. Now these viruses actually, they survive only in the living plants or in the animals. They produce their own proteins with the help of the host cell in which they are found, and thus they reproduce at a very faster rate. They replicate at a very faster rate, and then they destroy the host cell and they become free. They come out of the host cell and then they again infect the new cell. And as I said, you can see the tomato wilt virus, the virus which has affected the tomato and it has caused the spoiling of the tomato. There are many different types of viruses which cause disease in plants as well as animals. Like in human, poliovirus, influenza virus, HIV, AIDS. In cattle, it is picorna virus. In plants, tomato wilt virus and tobacco mosaic virus. And in bacteria, bacteriophage, which attacks bacteria. And you know what corona is? It is also a virus. So it is also having DNA or RNA and it is covered by a protein coating, right? So students, this is all about the first lesson, that is living world and classification of microbes. It was quite a short lesson and we had finished it in this one single period. So students, next time please be ready with the question answers of the workbook. If at all the books are not distributed or if you are not able to get the workbooks, please keep your textbook and a notebook ready so that you can just jot down the page number and paragraph of the answers of the workbook exercise. Though the school has already distributed the books as per the schedule and the second round of distribution of books has also taken place in the last week, I think from 9th to 12th. So students, I hope you all have collected the books from the school. If not, please collect as soon as possible as per the schedule given to you on your WhatsApp group and the app, school app. And students, please take due precautions. Please don't go out of your house if there is no emergency. Please take care of your health. Take care of your elders, your parents, your relatives in your house, and stay at home. Stay safe. Take care. Thank you.